Hello everyone, my name is Dr. Daniela Franca, and I'm going to talk to you guys about the historical role of animals in the emergence or maintenance of infectious diseases of people, commonly known as zoonoses. So what are emerging infectious diseases? Well, according to Morse in 1995, emerging infectious diseases are infections that have newly appeared in a population or have existed previously, but are rapidly increasing in both incidence or geographic range. Why is it important to know about them? Well, on one hand, they can cause high numbers of morbidity and mortality cases, such as the one seen with Spanish influenza, a pandemic involving H1N1 influenza virus, which resulted in 500 million infected people and 50 to 100 million deaths. They can cause huge economic losses, especially when livestock and poultry is involved. For example, in foot and mouth disease, losses arise from the direct effects of the disease on production, from the cost of disease control, and the restriction of trade. In highly pathogenic avian influenza, the poultry from the affected country becomes worthless on the export market. They are also at risk of being involved in bioterrorism. This is actually a very serious threat since generally the agents used result in high mortality rates and major public health impact. Examples of agents that pose a risk include anthrax and plague or Yersinia pestis. They can cause social disruption of activities such as trade, travel, and businesses, which can create a major socioeconomic crisis. And they can cause loss of biodiversity. What is interesting about this is that it can go both ways. So emerging infectious diseases can cause loss of biodiversity, such as the one seen with chytridiomycosis in amphibian population, or white nose syndrome in hibernating bats. A lot of the populations of this type of species have almost gone to the risk of extinction. But loss of biodiversity can also affect the risk of infectious diseases exposure by increasing pathogen transmission, allowing close interaction between species, and increasing disease incidence. So what are the precipitating or driving factors for infection disease emergence? Well, the factors that have contributed to the increasing rate at which diseases have emerged in recent years include ecological changes, both natural, like climate change, or anthropogenic drivers. Within the anthropogenic drivers, we have agricultural or economic development as a response to the overpopulation crisis. Population movement and commerce, particularly starting in the late 20th century. So population movement can be due to wars, travel, or migration. So for example, workers moving from rural areas to the cities. Obviously, due to globalization, new transports were created, buses, cars, trains, planes, allowing mass transportation of not only people, but pathogens as well. And this obviously sped the dissemination of diseases. Changes in human behavior. An example of this is seen with human immunodeficiency virus, with the continual use of, for example, intravenous drug or the acts of sexual intercourse. Technological development in both industry and health. So for example, in the food production industry, as a response to the overpopulation, there is an increased demand of food. Operations are forced to increase efficiency and reduce costs, which unfortunately increases the possibility of accidental contamination. And pathogen adaptation and change. So basically, this is in response to the wide and sometimes inappropriate use of antimicrobial drugs and even other chemicals. This leads to the pathogens developing resistance to drugs. Here we can see some of the major factors that underlie disease emergence and reemergence. They can be either respective of the agent, the host, or the human environment. This map shows examples of emerging and re-emerging infectious diseases, which can be seen pretty much in every part of the world. By newly emerging diseases, basically those are the ones that are recognized in the human host for the very first time. Re-emerging infectious diseases are diseases that have historically infected humans, but continue to appear in either new locations or in drug-resistant forms, or that reappear after apparent or controlled elimination methods that didn't work out exactly as they planned. However, what's very important to note is that 75% of emerging infectious diseases are zoonotic and originate mainly from wildlife species. Today, I will talk to you about these 4 diseases to demonstrate the interactions between animals and people and how these interactions led to the occurrence of zoonoses in the past. These 4 examples include human immunodeficiency virus or acquired immunodeficiency syndrome, Nipah virus, Lyme disease, and bovine spongiform encephalopathy. Human immunodeficiency virus or acquired immunodeficiency disease syndrome. Well, a little bit about the historical concept. It was only recognized as a specific disease in 1980, and the human immunodeficiency virus was not identified as the etiologic agent until 1983. It originated in Africa. There was actually a cross-species infection of humans by the simian immunodeficiency virus. The virus was associated with the acquired immunodeficiency syndrome. There are 2 types of virus, so there are HIV-2, which is found in sooty mangabes, and HIV-1, which is found in chimpanzees. Both are retroviruses. Approximately 16 million people have died from acquired immunodeficiency syndrome, and 50 million are currently infected. Transmission can be either through vaginal or anal sexual intercourse, contact with contaminated blood, or perinatally from mother to child. So basically, 2 theories have originated explaining how the virus managed to jump from primates to humans. One of those theories is Edward Hooper's theory. Basically, he was a BBC radio reporter. In his book The River, he explains that the acquired immunodeficiency syndrome epidemic was caused by a contaminated polio vaccine that was given to hundreds of thousands of people in Central Africa in the 1950s. He showed the strong correlation between the rural African clinics where the Wistar oral vaccine was administered from 1957 to 1960 and the villages where the earliest acquired immunodeficiency syndrome cases appeared. You guys can see it over here. So basically what happened was this. A pure strain of poliovirus was isolated. The poliovirus was grown in a series of monkey kidney tissue cultures until it was incapable of causing diseases. The weakened strain were tested on chimpanzees to ensure that they were safe and effective. It is then grown in large quantities in fluid from over a single layer of monkey kidney tissue to produce the vaccine. Finally, the vaccine is administered orally to humans to trigger the immune response. However, this is what happened. Apparently, the kidney cells that were used to grow batches of the vaccine might have come from monkeys infected with the simian immunodeficiency virus. The second theory was that of Preston Marks, a virologist from University Davis. He contends that the AIDS epidemic started with the reuse of contaminated needles during disease eradication campaigns in the 1950s in West and Central Africa. The needles turn a harmless monkey virus into HIV through a process called serial passage. So how does the process work? Well, let's say we have a market woman that is infected with the simian immunodeficiency virus after she butchers at sooty mangabe during food preparation. Let's remind ourselves that in Africa, hunting takes place in order to get food. She then receives an injection. That same needle that was used on her is used on a second person, so the simian immunodeficiency virus from the woman infects the second person. That second person later returns for another injection with a new needle. A third person comes in, and that same needle is reused on him, infecting him. The simian immunodeficiency virus starts to adapt to the human immune system and grows stronger. That third person that came receives another injection with a new needle. Yet again, that same needle is used again on a fourth person. Keeps going. The simian immunodeficiency virus adapts and grows stronger. The fourth person returns again for another injection. Again and again, the virus continues to passage through several more people through the reused needles. Eventually, the simian immunodeficiency virus transforms into HIV and is virulent enough to pass through other means. Basically, the simian immunodeficiency virus was first isolated in 1985 after the virus infected and killed Asian monkeys in a US research labs. Okay, so how did the disease disseminate and become a pandemic? Well, it first obviously had to jump the species barrier. So it could either have explained by the two theories presented, so both Hopper and Marx. Another possible explanation is the hunting, preparation, and consumption of wild animals. So hunting of non-human primates for food does take place in Central African forests. These animals that were hunted were then brought to the market to be butchered for food. So the possibility of the virus crossing the barrier through a bite or a hunter's contact with contaminated blood on an open cut or injury with the monkeys when the monkeys were butchered for food is quite a possibility. There's also the fact or the presence of the pet market. So orphan infant manga babies were kept as pets. And in relation to the dissemination, well, human migration and travel took place. So due to the social upheaval in Africa during the independence struggles between 1950 and 1960 and subsequent urbanization, roads were built through the rainforest and colonial travel prohibitions were lifted, and this enabled HIV carriers that had previously lived in isolated area communities to migrate into the cities, where they spread the virus along trade routes in Africa. It was then spread regionally along highways, and then by long-distance routes, including air travel to more distant places. There was also obviously the changes in human behavior, so commercial sex work through either heterosexual and homosexual transmission, and intravenous drug use. Actually, the prevalence of AIDS was seen among African soldiers and prostitutes. Then the development and innovation in healthcare, so in relation to blood transfusion and blood products, and the development of drug resistance and ability to avoid the immune system from the HIV virus. Nipah virus. So a little bit about the historical context. It belongs to the family Paramyxoviridae, genus Henipavirus. In 1999, it was first isolated from pigs and pig farmers in Malaysia. In 2001 occurred an outbreak in Bangladesh, and from there, nearly annual outbreaks are seen. There's a periodical identification of an outbreak in eastern India. Other regions at risk of infection include Cambodia, Ghana, Indonesia, Madagascar, the Philippines, and Thailand, due to the presence of the natural reservoir of the virus, which is the Pteropus bat species. It has a high case fatality rate on average of 73%. As mentioned before, the Pteropodidae family fruit bats are the natural host of Nipah virus. And outbreaks have been seen in other livestock and domestic animals during the initial Malaysian outbreak in 1999. So how does the transmission take place? Well, Nipah virus can be transmitted in three ways, either through the consumption of fruits or fruit products such as raw date palm juice contaminated with urine or saliva from infected fruit bats, direct contact with infected pigs who get infected after they are exposed to bat droppings, or direct human-to-human transmission. So here we can see how livestock and wildlife are involved in the occurrence of disease. What were the factors that promoted the interaction between animals and people, potentially leading to the occurrence of Nipah virus?